So welcome everyone to the third installment of the PhenoTips Speaker Series titled The Importance of Deep Phenotyping in Precision Medicine. I'm your host, Dr. Paweł Buczkowicz, and joined today by our very special guest, Dr. Peter Robinson. Thanks for joining. The agenda for today's 60-minute webinar will include some introductions and then a 45-minute presentation by Dr. Peter Robinson, followed by a question and answer session. You can submit your questions through the chat or through the Q&A function in this application. If you use that feature, you can also upvote questions that are posted throughout the presentation to make sure that the most popular questions get prioritized. This webinar series is brought to you by PhenoTips, the world's first genomic health record system, a software and service challenging the status quo of genomic medicine by modernizing and streamlining your workflows. It includes features like pedigree drawing, standardized clinical symptom capture in HPO, and diagnostic insights. Since electronic health records aren't built for genetics, PhenoTips fills the gaps by providing a complete suite of digital genetics tools. In light of the pandemic, and many of our colleagues, collaborators, and friends working remotely from home, PhenoTips is now hosting this speaker series. So I'll be your host. My name is Dr. Paweł Buczkowicz. I'm the COO and VP of Scientific and Medical Affairs at PhenoTips. My drive and calling are actually to make tangible and beneficial impact on how medicine and healthcare is practiced. I do this through advocating for technological innovation, collaborating on cutting-edge research across the fields of computer science and medicine, as well as helping optimize clinical workflows and operations. This is why I am very excited to actually introduce our topic and speaker for today's webinar. Precision medicine promises to improve all areas of medicine, and genetics' increasing role in clinical decision-making is also highlighting the importance of data models, computation, and ontologies. Phenotypic analysis is an invaluable tool for genotype-phenotype correlation and variant analysis. Without computation data structures for phenotyping, this analysis would be time-consuming, variable, and often error-prone. Deep phenotyping with the Human Phenotype Ontology, which has more than 13,000 terms describing dysmorphologies, signs and symptoms, and over 156,000 annotations to hereditary diseases, is further supporting the drive for genomic medicine. With an ever-increasing role of genetic testing, long waiting periods for patients to consult with geneticists and GCs, deep phenotyping will play an increasingly important role in precision medicine. I'm honored to introduce to you Dr. Peter Robinson, who will present to you on the topic of the importance of deep phenotyping in precision medicine. Dr. Robinson received an MS in mathematics and computer science from Columbia University and an MD from the University of Pennsylvania. He completed training as a pediatrician at Charité University Hospital in Berlin, Germany, where he and his group developed the Human Phenotype Ontology, an international standardized vocabulary for phenotypic abnormalities encountered in human disease. Each term in the HPO describes a phenotypic abnormality, and the HPO is used by several world-renowned institutions and projects, such as the Sanger Institute, NIH-funded groups like the Undiagnosed Diseases Program, Genome Canada, the 100,000 Genomes Project in the UK, and of course, also the main ontology used in PhenoTips. His group developed algorithms and software for the analysis of exome and genome sequencing data and identified several novel disease-causing genes. He's published hundreds of manuscripts, many of them in prestigious journals like Nature Genetics, Cell, the New England Journal of Medicine, to name a few, and according to Google Scholar, has garnered over 19,000 citations for his work. He is currently a professor and Donald A. Roux Chair in Genomics and Computational Biology at the Jackson Laboratory in Farmington, Connecticut. He is also the director of the Human Phenotype Ontology Consortium and the co-program director of the Monarch Initiative. Dr. Robinson, please take it away.
Thank you. Thank you for the kind introduction. So we'll share my screen. Are my slides now visible?
They are.
So yeah, so I'd just like to start. Thank you for the introduction, and maybe start off with a little history that might be of interest to this audience. So we started working on the HPO in 2007, published it in 2008, and PhenoTips I forget actually the exact date when PhenoTips came out, but it was the first software that actually made it easy for people to use HPO to annotate patients, and was I think enabled a lot of people to start using HPO. So we've always been grateful to PhenoTips. I'd also say at the Jackson Lab, it's not a hospital, so I'm no longer involved in clinical care as I was in Berlin at the Charité. But we were using PhenoTips there, and I very much like the software. So I should say I'm not getting any money from PhenoTips, but if they want to buy me a beer as a result of this endorsement, I probably would accept it. Okay, I'm going to start now. So oops, why can't I now? Here we go. So I guess the first thing that people might ask, and we've heard this question a lot, is sort of what is an ontology and why do you need it? And so I usually start off talks with slides like this. So Margaret Dayhoff was actually the first bioinformatician, and she came up with matrices called PAM matrices that are very similar to the BLOSUM matrices still used today by the software called BLAST. And so if you're searching for a DNA sequence, you'll send the sequence to the server at NCBI, and you'll get, there's a very elegant algorithm, and it will calculate the best hits and return them to you. And so this is sort of the thing that most bioinformatics software does, or a lot of it. Sort of, you have something you're interested in, but you're just trying to find it, send it off to a database, and rank the similarity to elements in the database. And we were doing medical genetics in Berlin and trying to do the same thing with patients, but it's not immediately obvious. How do you calculate a similarity between a guy with a common cold, somebody with a broken arm, somebody with sepsis, or an EKG abnormality, and so on. And the answer that we came up with involved ontologies. And so let me just give you a little intuition about how ontologies work. So I'm not going to be very mathematical or algorithmical in this talk. It's all about intuition, but if you do have some questions, I'd be happy to answer. So this Tom Gruber was one of the giants in computer science, and he was working in the field of knowledge representation in the 90s, and he was the first one to come up with a really great definition of ontology. And he said it's a specification of a conceptualization. And so the first time I saw that, I was not enlightened. It's not, it's very concise, but actually it's easy. So a conceptualization, that means that you're talking about a domain of knowledge, and you just say, what are all of the concepts? What are all of the items in this domain? So for instance, if I'm talking about wine, which I do a lot, the items of your domain of knowledge might be Rioja, they might be Cabernet Sauvignon, they might be Riesling. All of these are very delicious wines, and you can imagine having a wine catalog that might have hundreds or thousands of entries, and that's not an ontology yet, but that's a conceptualization of the domain of wines. And if you provide definitions such as Rioja is a Spanish red wine, then you get a glossary or a dictionary. And if you define synonyms, then you get a thesaurus or a thesaurus. And so I don't think it's too common for wines to have synonyms, but obviously many items and many domains have long lists of synonyms, especially medicine. None of this is an ontology. When you start to specify how the items in your domain are related to each other, then you get an ontology. So everything on the right side of the red line. And the most common way of specifying how items are related to each other is subclassing. So Rioja is a subclass of red wine. And red wine and white wine and rosé wine are all subclasses of wine. So you get a hierarchy. And so that's an ontology. And there are many, many different kinds of relations between objects. So instances, for instance, Mickey Mouse is not a subclass of mouse. Mickey Mouse is an instance of mouse. And let's say nucleus, it's not a subclass of cell. It's located in cell. And so I'm gonna skip some of the details, but it's a very beautiful part of computer science, and and we've been working on ontologies really in the field of gene ontology for some years, and basically decided, well, let's start to apply this to the human phenotype. And in the past 10 years, we've all seen this sort of explosion of big data that's now starting slowly to be used for medicine. And so if you're interested in reading about that, we published a review about 2 years ago that gives sort of an update on not just on HPO, but just on what ontologies are being used in medicine and where we might go. And so this is sort of a vision slide, but we're defining Fanconi anemia, which is on the left side of the slide, basically by sort of saying, well, what characterizes this disease? So has phenotype points to a list of terms that you can see, the orange ones that say microcephaly and short stature and so on. Those are HPO terms. But you might also want to define the disease based on how you can test for it. So you can, with Fanconi anemia, you can test for DNA damage, chromosomal breakage. There are environmental factors that influence this hereditary disease, such as aldehydes, or you should not give cross-linking agents to these patients. It's got subtypes that are related to the genes. So Fanconi A, Fanconi B, and so on are individual genes which, if mutated, are associated with this disease. And these genes encode proteins that are members that fulfill certain functions in the cell. Many of them are involved in DNA repair. And I guess the interesting thing is that there are intersections between different diseases. So for instance, acquired aplastic anemia has a very similar sort of blood phenotype to Fanconi anemia, but it doesn't have the malformations. But some of the risk factors are roughly similar, so they can also damage DNA. And I think it's quite likely that as we start to understand more about molecular medicine, that just analyzing the phenotypes and environments and will actually help us to understand how to stratify patients for treatment. And I'm not going to talk about this today, but the Monarch Initiative, it has been developing ontologies for environmental exposures, for medical actions, including treatments, and some other things that actually go part of the way to fulfilling the vision shown in this slide. So that's sort of the sexy part of ontologies. The boring part is shown here, and that's just plain old interoperability, and that's important because when I was starting to work on genetics in the 90s, there were lots of boutique databases about genetic diseases, and each of them was using its own separate schema, and so you just couldn't exchange data between databases, and it would have been impossible to analyze an exome where you really need sort of the same standards for each gene in the genome in the 90s using the databases that existed then, because they were just all different. And so Tony Brooks, who's one of my favorite colleagues, and we wrote a paper about databases, and I contributed, basically some sections about how ontologies are used to help connect between different databases. And so this was sort of the situation in the 90s. Well, actually, still even in this millennium. And if you're talking about the same thing, you can find very different descriptions. So if you're talking about muscle wasting that affects many different muscles, the medical term is generalized amyotrophy. But it turns out there are at least 10 synonyms that you can look up in the corresponding HPO terms. And so a computer doesn't know that generalized amyotrophy is the same thing as muscular atrophy, generalized. And that's where just basically having an ontology, can help to connect data from different sources. And so now I'm going to start to talk about the HPO. So this is one of the terms of the HPO. It's for macrocephaly, which basically just means a large skull. And each term in the HPO is identified by an accession number. These are these HP colon 7-digit number that you may have seen. And we have tried to provide an English language definition for each of these terms. And our goal is that, postdocs, doctors from different fields can understand it. So, a radiologist should be able to understand the definition of an immunology term in the HPO. We provide lists of synonyms that we think are useful for text mining and just finding the terms. And we do provide cross-references to other ontologies. There are a few other things in the each term that I'm going to skip for right now, but might get back to it. So this is just a representation of some of the work we've done on HPO, and I'm showing this not to say how many publications I've published, but to say we have published, because many of these publications have had 50 or 70 authors, and these have been clinicians who have helped us to extend the HPO, and it's become quite a community project. And so for the past 12 years, we've been having 1 to up to 4 workshops a year. So one of the recent ones, for instance, was on eye disease, and we were very lucky that an eye doctor from Manchester, Panos Sargionotis, organized a workshop in the European Rare Disease Network, and about 30 ophthalmologists came to a wonderful place in France, and we basically went through the eye part of the ontology for 2.5 days and just added terms, fixed a few mistakes, and so on. And so that's a reason why I think that separates HPO from many of the other clinical terminologies, that we think the HPO represents, or tries to represent, really the state of the art of experts in the field. And it also has a much deeper and broader coverage of phenotypes. So currently about 15,000 terms, and the United Medical Language System at the National Library of Medicine actually did a comparison a couple of years ago and said if you took everything else that you have in their system, including SNOMED, and just said how many phenotype terms do we have, it was about 25% of the HPO. And I think the difference is, if anything, has only grown. And that's meant that UMLS has now imported HPO, and we've also, I'm not sure if this is public yet, but we've agreed to work with SNOMED to create a mapping which we hope will help those people who are starting to work with HPO terms in the electronic health records. Another thing that I just mentioned briefly that differentiates HPO from some other vocabularies is that each of the terms is defined with reference to other ontologies using logical expressions that are technically called OWL class definitions, but I'll skip the details. But so if you're saying deeply set eye, essentially you're talking about an eye. And so there's an ontology that has terms for each of the parts of the body. And then you're saying, well, what's the quality that's abnormal about the eye? And we're using, there's another ontology of qualities called Pato that we're using. And so by sort of making definitions such as that, we can define a lot of the things that are a lot of the HPO terms with references to abnormal functions in gene ontology, abnormal anatomical configurations. We can reference abnormal cell types. We can reference abnormal biochemistry, pathology, embryology, and a few others. And so this is one of the things that makes the HPO also a very sophisticated computational tool that a lot of people have exploited for algorithms, for instance, for genomic diagnostics. So just briefly, what does phenotype mean? Well, it actually means different things to different fields, to different people. And so, if you're a biologist, a phenotype is, let's say, the color of a butterfly's wing or the weight of a whale, whatever. But in medical contexts, it's usually a deviation from some norm. And another thing to realize about the word phenotype is that it's used in some contexts to refer to a disease, and in another context to refer to the individual features of that disease. And that's actually what we mean when we say phenotype. So we actually mean phenotypic feature. And that can be a sign. So that's something that a doctor will discover on a physical exam. So something like a heart murmur. A symptom is something that a patient complains about, lab findings in the blood or otherwise, imaging studies like chest X-ray or CT, neurophysiology, many other things like that. So this enables what many people call deep phenotyping, which is sort of the opposite of a Gestalt diagnosis. So if most of us were to see a child with Down syndrome, we could just say, oh, I recognize that Gestalt. You don't have to think very hard. But there's so many different diseases in genetics and in medicine in general that no physician can gestalt all of the diseases. And so most physicians consciously or unconsciously sort of record the individual phenotypic features and then try to do pattern matching. And so that's really what HPO is trying to support. And so one of the big things that HPO tries to support is genomic medicine. And when I was starting genetics in the 90s, the thing was somebody would come to you and they would have a disease such as neurofibromatosis, and well, you knew what gene it was. And so you would basically do a PCR of that gene and maybe screen that gene using, there were many methods that millennials will not ever have heard about, but if you found a nonsense mutation in that gene, you had made the diagnosis because you already knew what gene it had to be based on the clinical diagnosis. And that still is done today for some diseases. But for exome and genome sequencing, you don't know what the gene is. And so you have two challenges. One of them is to basically find the right gene that could be mutated. And the second is to decide, well, is there a truly pathogenic mutation in that gene? And the issue is that with typical bioinformatic filters, you're left with up to 500 candidates per exome, maybe hundreds more in a genome. And even if you have a trio or some other way of narrowing this down, you typically still have a list that's relatively long. And so one of the things that phenotype analysis can help you with is to take the list that you have for your exome or genome sequence, and it might be 5, 10, or it might be 500, depending on how you've done your exome sequencing, and basically to say, let's rearrange the list of candidates such that the candidate genes and diseases most likely to be the cause of what you observed in your patient are at the top of the list. And this means that when you're showing the result to a diagnostician, to a clinician, hopefully they just need to look at the first page and they say, oh yeah, that must be it. And then, they go about and confirm it by co-segregation or other analyses. And so this slide here is an example of why this is so difficult, and, well, at least in some cases. So this was when I was still in Berlin, and we were seeing patients, and I was fortunate enough to be in a department with some really good clinicians, and I'll mention Denise Horn and Rickard Neumann as two of the ones I had a lot to do with. And anyway, we saw these two patients within a few weeks of each other, and we didn't even realize that they had the same disease. And it turned out that they had something called Wiedemann-Steiner syndrome, which had recently been described as being caused by mutations in a certain gene. And I don't want to talk about that disease, but let's just look at these profiles. So the blue boxes show essentially the part of the HPO database, which is derived from OMEM and derived from the literature. And so this is sort of the standard profile of disease. And the yellow ones are for the girl and the red ones are for the boy. And we see, for instance, in the literature, we have short toe, that's the top line. The boy had long toe. The second line, short middle phalanx of finger. Well, the girl had cone-shaped epiphysis of the phalanges. So it's not quite the same thing, but it's the same organ. The neurological phenotype was similar, but the girl had microcephaly, the boy had macrocephaly, and there were a few other differences like that. And so, if you try to do an exact match in genetics, you'll fail. So you have to to be able to do a fuzzy match to even match the right disease. And the other thing that we found that you need is to weight the terms by specificity. So for instance, many patients will have intellectual disability. That's a very common feature in genetic disease, but many fewer will have, let's say, cone-shaped epiphysis of the phalanges of the hand. And so a lot of the software that uses HPO uses something called information content to assess the specificity of terms and to weight it. And one of the tools we've developed is called Exomizer. And so Exomizer will take as input a variant call format file, a VCF file that represents your exome or genome, and will also take a list of HPO terms. And essentially will filter the exome or genome for predicted pathogenic variants. And so if we look at the left side, and maybe you see the arrow underneath variant score, there are a bunch of stars representing variants, and two of the stars are dark. So those are predicted to be pathogenic by our software. And our software does a couple of really clever things, and it's going to be doing more clever things soon, but a lot of this is standard. So you look this up and look at polyphen or SIFT scores, and more recently CAD and Revel. But that all of that is, I'm just going to say, that's standard bioinformatics. That's what still leaves you with 10 or 100 candidates. And then we use the HPO terms that you've entered to calculate phenotypic profiles. And so for each of the genes, we associate it first with human diseases if available, and if not, we can map many of the genes to their orthologs in mouse and fish. And I'm not going to explain it right now, but we can match phenotypes across species. And there's a couple of other bells and whistles that I'll skip right now. But in this example, there's a good phenotypic profile for two genes, the one on the far right and the one on the far left. And if we combine the variant and the phenotype score, there's only one gene that has both a good phenotype profile and a good variant profile. And that's the one that Exomizer ranks the most highly. And so in the meantime, Exomizer has been used by quite a few people and consortia. And one of the groups was sorry, I can't right now. The NIH Undiagnosed Diseases Program, which UDP, and they looked at several hundred patients and basically said that Exomiser increased their diagnostic yield by up to 20%, and which is pretty good because they spend up to a week per case. And more recently, the 100,000 Genomes Project has been using Exomiser in its genomizer mode, so it also looks for non-coding mutations, and that it turned out to be the best performing software of quite a few softwares that were tested. So here are some numbers. There is a paper now under review that Damien Smedley and Jules Jacobson have been leading, and that will be out a little bit later this year, describing that experience. So I think I'm just gonna skip this for now because I feel like I'm possibly pushing the time. I'd like to talk about another software that we just published called Lyrical, and so I think this is a very interesting advance, and so if you look at studies in the detection rate for exome or genome sequencing is at best 40 or 50%, and in some cohorts it's even less than that. And so that means that in about half of the cases, and maybe even 3/4 of the cases, the disease or gene at rank 1 is wrong. And so Exomiser doesn't really help you realize that. It just sort of has something that's on rank 1. And so that's one thing we were trying to address. How do we estimate how good the matches are? And the other thing is that the way our software has worked to date, it just says, well, this is the phenotype matching score, but it doesn't tell you how well individual HPO features support a diagnosis or argue against it. And so our goal was to develop an algorithm to help with both of these things. And this has been basically my pet project for the last 6 years, and very happy that we managed to publish this this month, just a few days ago. I'll give you the reference in a bit. But this is what you would see if you're using the software. So in this case, we've entered a list of about 10 HPO terms that are shown on the spark lines under the profile match. I'm not sure if you can see my mouse, but you see the little green and red bars. And so in this view, we see that the congenital variant of Rett syndrome has a pretty good phenotype match for most of the terms that we entered. And for the last 3, it's not so great. And the last bar here is for the gene match, which shown on the very right, FOXG1 gene. And so this has a post-test probability that the software estimates as 100%. I mean, it's like 99.999 something. And the next candidate, the next couple of candidates still have a decent post-test probability. And if we look at the spark lines, we say, well, they all have some phenotype terms which match, but there are many that don't match. And as soon as we get down to the candidate at rank 5, you know, it kind of looks like it's just noise, and the post-test probability is estimated at 0%. So if you were using the software, my advice to you would be look at the first 4 candidates closely and try to figure this out. Once you get down to a post-test probability that's close to 0, essentially this program is not going to help you anymore, and you should probably, if you still want to search harder, you should use another program or another way of looking at the variance. But our program is basically saying, look at the top 4, especially the top 1. Sorry. And so this software is actually using a new mathematical framework. So as far as I know, all of the software that's out there that uses HPO is using something called information content. And this is something we developed in the Phenomizer algorithm 2009. And this led to these two problems that I mentioned at the beginning. And so this is a new framework, and essentially we're calculating a likelihood ratio which is, I think I showed, it's basically defined as the probability of seeing a certain abnormality given you have the disease divided by the probability of seeing that abnormality given you don't have the disease. And so the software, essentially we came up with a way of calculating that probability, and I'll refer to the paper for the mathematical details, and just say that we have essentially different equations for different configurations of query term and disease terms, annotated terms. And so we did a bunch of evaluations, and we basically wanted to compare this to a lot of other software, but a lot of the sort of HPO software that gets out there is published but not really supported, the academic software, or there's some very nice commercial software, but we can't really use that to test because we don't have a license. So we basically compared this to our own software, Exomizer, and if we're looking at the original data from 384 case studies that we biocurated from the literature, Lyrical had a very slight advantage, but essentially it's about the same as Exomizer. However, Lyrical has a couple of features that make it robust to noise, and we tried here in panels C and D to say, what about the case where you have an autosomal recessive disease, but you only pick up one heterozygous mutation? And so in short read exomes or genomes, sometimes you'll miss, let's say, a deletion of a single exon, right? So it'll just look like it's a reference sequence. And if you have a point mutation in another exon, then you might, the call that you'll get from GATK or whatever will be heterozygous mutation, but you say, oh, it's an autosomal recessive gene, so that can't be it. So a lot of software will not weight those hits very highly. But actually, if it's a really good phenotype hit, you still want to look at it as a clinician and maybe decide whether to look at the BAM file to search for a deletion, or just to do PCRs if you think it's a great candidate. And so Lyrical is using a genotype model that helps you pick that out. And this panel D essentially shows that even if we remove one of the two alleles, still most of the candidates get ranked at rank 1. And the bottom line is essentially just showing if we add noise terms, so we replace an original term with just unrelated HPO terms, how well do we still do? If we decrease the specificity of HPO terms by replacing them with a parent or a grandparent term, how well do we do? And here, in both cases, we're showing an advantage over the algorithm we're currently using in Exomizer for the phenotype matching. And so this is, if you use the tool, this is what you would see. It generates an HTML page for each of the top candidates and shows the contribution of each of the terms. In this case, we see that most of the terms are a good match, a couple are bad match. And looking at this, if you're saying, okay, this is Rett syndrome congenital variant, and we're seeing inability to walk, delayed speech and language, hypoplasia of the frontal lobes, well, actually these do match the disease. They're just not in the HPO database, which is nothing is complete in this world. So if you're looking at this as a doctor, you would say, well, yeah, actually, this is an excellent match for that disease. And yeah, that's it. I'm gonna talk. Do I still have time, Pablo? Yeah, I'm gonna talk about a couple of more recent things that go in direction of precision medicine. So we've been working with the Global Alliance for Genomics and Health to develop a format called PhenoPackets. I'd like to point out that many of the original ideas were developed by Ian Buske, who I'm not aware of his exact role in PhenoTips, but I'm sure everybody knows him. But we've expanded this, and Orion has continued to work on this with us, and to provide a general way of exchanging phenotype data for research or diagnostics. And the basic idea is um, you might be asking, well, wait a second, I thought you said HPO was a standard, but well, what if you're trying to, describe a patient? Do you send me a list of HPO terms in an Excel file? And if so, what about the age of the patient? And, what about things like severe or what if you excluded an HPO term? How do you do that? And there was no format for that. And so PhenoPackets does that. And it also has a lot of other elements that are intended for use in cancer and for common disease, for describing treatments. So the initial version of PhenoPackets was adopted by GA4GH last year, and we are now in the process of developing a version 2, which will have some extensions. This version is now being considered for ISO certification, and we're hoping that it will find a broad acceptance. So why would you want to use a PhenoPacket? Well, we think there are a lot of use cases. One obvious one is for diagnostic tools. And so, for instance, if you have phenotype data in phenotypes or something like PhenoTip, sorry, and you say, well, I want to use this for Exomiser. Well, currently, the format of Exomiser that existed until very recently was a YAML file, and you would have to either create this with some other software tool or pull out an editor and do this by hand, and it was kind of annoying. And so in the version of Exomiser that's being released as we speak, or this week or next week, you will be able to use a PhenoPacket as an input format. And so you could essentially say, export this from your software of choice and just run it. And so that would save a lot of time. Also, there are a lot of different phenotype tools out there that use HPO, and to date, each one of them has its own input format, but actually now Exomiser, Lyrical, and a tool by the Kai Wang Lab, who is at Children's Hospital in Philadelphia and doing really great work. And it's called, I think, Phentogene, also, I think, is using PhenoPackets as an input format. So we're hoping that that will just make it easier, so that if you say, I want to try these 10 tools because I wanted to see what they all say, now you can just use one input format. So that's obvious. Another thing which is really difficult is doing genotype-phenotype correlation studies. And so, when I was starting to do research in the 90s, my mentor said, let's just do genotype-phenotype studies on the NF1 gene and just look at some papers. And so the only way of collecting this was in something like Excel, which was just a complete nightmare. And essentially, journals there's no standardized way of capturing a case report from a journal. And so now we're actually talking with a number of journals in our field about PhenoPackets, and in a project led by Melissa Hendel, my Monarch colleague, we have funding from NHGRI to develop PhenoPackets for journals. And so maybe the next time one of you submits a case report, you'll be asked if you want to submit a PhenoPacket, and that will go into a databases. Similarly, for patient registries and trials, and we think there's actually quite a few use cases of just saying this is a standard way of representing the data, so that anybody who's writing algorithms will know where to start. This is going to be my final topic. I'm not able to see the clock because I have a full screen here. So if I'm starting to run out of time, Pavel will start to wildly gesticulate, I hope, so that I see it. But I'm going to start talking about electronic health records and precision medicine. So everything to date has been about rare disease. Now we're getting into the area of common disease. And so in electronic health records in this country, most hospitals use LOINC to denote lab tests. And so currently, actually it's more than, I think it's closer to 100,000 now. There are 100,000 different LOINC lab tests. And so if you're getting this information out of an electronic health record, you'll have this LOINC number, and you'll have a number that represents the outcome of the test. And we were starting we were participating in a project where people were doing machine learning on EHR data, and they were just taking the results from lab tests and essentially making one column for each LOINC code. And we thought, well, actually, that's kind of stupid because many of these LOINC codes are very related. And so, for instance, nitrite in urine, that's something they'll measure if you are suspected of having a bladder infection. And if you do have nitrite in your urine, you probably have a bacterial bladder infection. And you can measure this with a strip. I'm sure you guys have seen that. You can also put this, put a drop of urine into a machine, and there are several varieties of each. So there are actually 4 LOINC codes for this. But if any one of these tests is positive, you have nitrituria, and the medical interpretation is the same. And so we developed a software library in Java that essentially takes the LOINC code, the outcome of that code, which can be represented in several ways, including something called Fast Healthcare Interoperability Resource, or FHIR. And so if you're doing a potassium test and the result is low, you have hypokalemia. And so just to give you an idea of how you might use this, we tested a cohort of about 16,000 patients at University of North Carolina who had asthma or other basically respiratory diseases. And we were able to map, I think about, getting how many, I think it was like 60 unique codes per patient. And we basically just said, which of these codes are predicting asthma? And for most of these codes, for some of these codes were just mapped to one LOINC term, but you see roughly a third of them were mapped to 2 or more than, even more than 3 or more LOINC terms. And so, whoops, I think I'm missing a slide here. Anyway, we managed to, we did a very simple statistical analysis with logistic regression and found 5 HPO terms associated with asthma, such as eosinophilia. And actually each one of these corresponded to a published study about an asthma biomarker. And we didn't have anything that was not published. And so this is a pretty impressive result for a pilot study. And this is, we're now using this actually in a large project called the National COVID Cohort Collaborative, which is being led by my colleague Melissa Hendel and by Chris Shute at Johns Hopkins. And this is going to be the largest collection of data ever, really, electronic health record data on one disease brought together. And so we are anticipating data on several hundreds of thousands of patients. And this is a big project. I'm not going to go into all of the details. A paper is just coming out in JAMIA shortly about it if you're interested. But what we're doing is we're basically transforming the data that is coming from this project, which is encoded using one of the EHR formats called OMAP, into one phenopacket per patient. And we've been developing machine learning software to essentially learn from graphs of data and texts of data that we are going to be applying to this. And this is called Embiggen. This implements something called Node2Vec, which was published about 5 or 6 years ago by Google. And we actually managed to speed it up by a factor of 1,000. I love saying that. And that this actually enabled us to, and you know, previously using the software from Google, this would take, you know, 36 hours on, even on powerful software. Now it takes, you know, 15 minutes for one run. And so that enabled us to optimize the parameters and actually boost the accuracy, the ROC, by about 15%. And we've Justin Rees, who you see here, has been one of the colleagues. He's in Chris Mungel's group, who's also in Monarch, on a first version of a knowledge graph about COVID-19 that you might also be interested in. Anyway, I think that's about all I want to say. I want to thank the many people, only some of which are listed here, who have contributed to HPO over the years. So we've been really blessed by the amount of interest in the community and the contributions that we've been lucky enough to get. So thank you for listening.
Thank you, Peter. We'll jump right into the questions, as we've had quite a lot of interest come through. And one sort of relevant to your last point about a lot of the contributors that you can see on this slide. Somebody asked, what is the process for updating / adding HPO terms to the database? So how can other researchers and clinicians contribute to the HPO?
Yeah, so there are two ways. So if you want to contribute one term or have noticed one error somewhere, then we have a GitHub tracker. And so if you go to the HPO webpage, there, maybe I can just show, can I show? So if you go to the HPO webpage, there is some documentation about this. And so if you go to how to contribute to HPO, there's a little bit more information about this, but the main way for one term is to go to GitHub. And so we've actually had over 5,800 requests over the last, I think, 8 years here. So it's been pretty active. And we're still working on about 200 of them. And you would basically say new issue. And so if you wanted to say, I suggest a new term for something, and you would say what the name of it was, and you would provide some information here. And so that it's described on the website if you want to do that. So we very much appreciate contributions like that. If you're in a consortium and you're studying, let's say, you know, spleen disease, and you say, actually, you know what, the HPO needs about 50 new terms about the spleen, then the best thing to do would be to get into contact with me by email, and we would set up essentially a small workshop. And we used to do these in person, but who knows what we'll be able to do in these interesting times.
Certainly, yeah. So we obviously encourage people to submit terms to the HPO, because the more collaboration we have from a variety of specialists, the better the ontology gets, certainly. We had a few other questions come through. We have, so somebody said, while working with the HPO data in data science, we regularly encounter the problem that there seems to be a significant intra-observer bias, because the HPO terms can be very specific or can be very broad. So they're saying next to that, because there are so many, sometimes terms are alike, while sometimes, while not being in the same, I guess, tree of the ontology, or in the same, the same part of the ontology. They said, how do you handle this? Are there any insights for that?
Well, yeah, I mean, this is a general problem that it has afflicted many people and many projects. There's no general solution to that, but it is possible to say, so if you have an annotation to something that's really specific, by inference, all of the ancestors of that term are also positive. So let's say you're talking about, abnormal cerebellum morphology. You can go down about another 6 levels in the HPO. But if, half of your clinicians just entered abnormal cerebellar morphology and the others went way down, you could just say, well, I'm just going to inherit everything up to the level of abnormal cerebellar morphology or not abnormal cerebellar morphology. That might be appropriate if you have very mixed levels of detail. Another thing to do, actually, I swear I've never gotten any money from PhenoTips, but it is a nice software, and you can set it up to say, everybody who's participating in this study needs to answer these questions, yes, no, or I didn't look. That's correct, Pavel, right? You can still do that, right?
Yeah,
So
that's correct.
that would be one way of sort of trying to ensure that everybody who's participating in your study is annotating to a required level of detail, and having, an explicit, no, I ruled it out, but is much better than just not entering something. And I think PhenoTips also supports that. So the best way is to get good data before you start to do the analysis. And the second best way is it's never a good idea, but it might be good to just sort of inherit everything up to a general level in that case.
Great. I think we still have time for one more question. So I'll sort of swing it towards the precision medicine side. What do you think are some of the barriers that may have been hindering the sort of the progress of precision medicine, and have any of those things changed in the last 5 to 10 years?
Well, I think it's just important to realize, precision medicine is something that's going to take, decades or centuries to realize. And, the war on cancer, so-called, was initially started by President Nixon about 50 years ago, I think. And if you read, there's a great book, Cancer, the Emperor of All Maladies, that just describes how the cancer research and treatment has progressed over the years. And, really it's over 200 years, small steps, but now we are so much better than we were back then. I'm pretty sure it's going to be similar. Maybe it'll be a little bit faster with precision medicine, but, I think one of the main barriers is phenotype. So getting sufficient data of sufficient detail from a sufficiently large number of patients, that's one of the motivations for phenopackets. So, in essence, you could now extract a relatively detailed representation of a patient from an electronic healthcare record into a phenopacket. And if you did this for 100,000 patients in your hospital and could correlate that with genomics data, that would, I think that would accelerate things. And so that's, if you are interested, the PhenoPackets project is GA4GH. It's very open, and we're now in the process of sort of finalizing version 2, but there's definitely room for ideas, contributions, people to test things out over the next 6 months or so. So I would encourage you to join us.
Right, we have time, I think, for one more quick question, but we probably have to keep the answer to about 20 seconds. So the question is, the software filters out the less relevant and less specific features in order to reach a differential diagnosis. Is it not precisely what the experienced clinician does?
Well, you may have noticed I have difficulties with verbosity, so it's hard to give a short answer to that. But yeah, I mean, with a lot of luck, any of these algorithms will simulate to a certain extent what an experienced clinician would do. None of them does it really very, none of them would come as close to a clinician who knew that disease already, who saw the patient there, saw the data. So I think there's still a lot of work to be done in bioinformatics to get the algorithms to work better and better.
Great. Well, thank you, Peter, for presenting for us today and for the insightful discussion as well. So you can follow Dr. Robinson on Twitter. The handle is PNRobbins. You can also follow the Monarch Initiative, the Human Phenotype Ontology, and Jackson Labs using so Twitter handle for Jackson Lab is Jackson Labs, Monarch Initiative is Monarch Init, and Human Phenotype Ontology is HP_ontology. Also, a big thank you to our audience today for tuning in and for asking those questions. You can follow us PhenoTips on Twitter and LinkedIn as well to stay tuned about the latest updates, events, and also the latest speaker series schedule. Please stay tuned for our upcoming October webinar on leadership roles in genetic counseling. You'll see a feedback link at the end of the session. Please click on it and take a minute or so just to answer a few questions, and you can vote on what future webinar topics may be. Thank you very much, everyone. Take care.
Thanks for listening. Bye-bye, everybody.